Hello everyone. In this particular class, we'll try to learn about what is differential leukocyte count and how it is performed. The aim of this practical will be determination of percentage of W basis. The principle is here, a blood film is stained with Leishman stain is examined for different types of white blood cells and the percentage of distribution of these cells is determined. The apparatus required are glass lights, Leishman stain, microscope, distilled water, lancet, and spirit swab. If you see the composition of Leishman stain, it contains eosin and methylene blue, which is dissolved in acetone-free methyl alcohol. So it should be acetone-free because acetone is a decolorizing agent and it is going to lyse the cell membranes, especially RBCs. The function of methyl alcohol is to fix the smear on the slide. The methyl alcohol precipitates the proteins which are present in the cells, and that precipitation attaches the cells to the cell smear. Eosin stains cytoplasm and basic granules, whereas methylene blue stains the nucleus and acidic granules. Eosin is a acidic dye and it is going to stain the basic granules, whereas methylene blue is a basic stain and it is going to stain the acidic part of the cell. To make a smear, take two grease-free slides with smooth edges and select one as a spreader. With aseptic precaution, prick the finger. Place a medium-sized blood drop near the one end of the slide. Do not touch the slide. With the left middle finger and thumb, hold the slide. Place the edge of spreader just in front of the blood and draw the spreader back until it touches the blood drop. Let the blood spread over the edges of the spreader. Then, maintaining appropriate angle 30 to 45 degree between the two slides, push the spreader to the other end of the slide by smooth, quick and uniform movements. Okay, so this is how the procedure is to make a good smear. So take a drop of blood. Okay, so then place the spreader just in front of the blood drop, then pull it back and spread the blood uh, throughout the edges. Okay, so then maintaining uniform forward movement, just push it in forward direction, maintaining 30 to 45 degree angle. Okay, so this is another diagram showing. Keep the spreader like this and pull it back, spread the blood uh, uniformly, so then move it in a forward direction with maintaining 30 to 45 degree angle. So characteristics of good smear would be the good smear is tongue shaped with smooth tail. It does not cover the entire area of the slide. It has both thick and thin areas with a gradual transition. It does not contain any lines or holes, that is any striations or windows. So these are some of the examples of correct and incorrect slides. It has many striations, it has windows, and this is a thick smear with the windows. Okay, and this is a nice smear with the head, body part, and tail, and there are no striations or windows. Prepare a second or third smear using the same procedure. Dry the smear quickly by waving the slide in the air. So again, note that the ideal smear has a thick area of head and gradually thin out at body and the tail. It's a tongue shaped with no windows or striations, longitudinal transverse. Then staining of the smear. Counting the drops of a Leishman stain, pour sufficient drops of stain on each of the slide to cover the whole smear and not to overflow. Then wait for 1.5 to 2 minutes for proper fixation of the smear. Note that during this period, do not allow the smear to dry, and if needed, you can add few more drops. Okay, so allowing them to dry it may lead to precipitation of uh, the stain. Then add the double amount of the distilled water, or if you have a buffer solution, add the equal amount of the buffer solution. Then mix the stain and water thoroughly by gently blowing air through the pasture pipette. Then let the diluted stain remain on the smear for 8 to 10 minutes. Okay. So this particular phase is called as staining period, and initial phase 1 to 1.5 minute is called as fixation period. Okay. So then at this stage a scum layer is going to appear, uh, so that indicates that ionization of the dye has taken place. Okay. Then later discard the stain and wash the smear gently under the tap water. The the tap water should not fall directly on the smear as it may be washed off. Let the smear dry in upright position. A well stained smear appears faint purple in color. Focus the smear first under low power objective to scan the entire slide, and then under high power for selecting the field. Then add a drop of cedar wood oil on the selected field. Lower end of the oil immersion till it is just immersed in the drop. So that means the objective of the oil immersion should touch the cedar wood oil. So then the smear is focused by using fine adjustment screw only. Then count 100 WBC by moving the slide in zigzag fashion or manner. Okay. So this is example of ideal smear, and this is example for ideal stained smear which appears light violet in color. So this is an ideal smear which contains head, body and the tail. Okay. And the morphology, the RBCs will be overlying in the head part, so you will not be able to appreciate proper morphology, whereas in the end of the tail the cells are too much spread. Okay. So in this part where the transition, the cells are properly spread and you can see the monolayer cell. Then the WBCs can be differentiated by the following points. The size of the cells. The largest cell the WBC you can see is monocyte, then probably large lymphocyte, a neutrophil, uh, slightly bigger than that is eosinophil, then neutrophil and basophil, then the small lymphocyte. Small lymphocyte is a similar size of that of an RBC. So then, depending on the features of the nucleus and number of lobes, neutrophils usually they are multi-lobed. The eosinophil is bilobed, whereas the lobe of basophil is S-shaped. Sometimes the lobes or nucleus are not visible. Okay. So then, features of cytoplasmic granules. Neutrophils have granules which are fine, and they're both acidophilic and basophilic granules, whereas eosinophils, their granules are very coarse and they are basically eosinophilic granules, and the basophil granules are also, they are very coarse and they they are basophilic granules. Okay, so then based on the nucleus and cytoplasmic ratio, uh, the monocyte has a 50% of the nucleus and cytoplasmic ratio, whereas in lymphocyte the nuclear ratio is high. Okay, so then when you are counting different WBCs, they can be denoted with alphabets. Neutrophil can be denoted by N or P, that is polymorphonuclear cell. Eosin is denoted with E. Basophil is denoted with B. Lymphocytes, whether it is small or large, it is denoted by L. Monocyte is denoted by M. Okay, so then the number of different varieties of WBCs are expressed in the percentage depending on their count. So this is how different WBCs they appear in the smear. Okay. For example, these two cells are neutrophils, which are bilobed, which contains fine granules, both acidophilic and basophilic. So this is eosinophil, which is has a bilobe nucleus, and it has a coarse granules which stained in brick red in color. So this is basophil, slightly smaller than the eosinophil. The granules are very coarse, and the nucleus will not be able to appreciate properly because of the overlying granules. So this is a large lymphocyte, okay, so where the nucleus is very uniformly stained, uh, and this is the monocyte where the nucleus is inhomogeneous, and almost there is a 50% of cytoplasm and nuclear ratio. Okay, and here the kidney is uh, the, the nucleus is kidney shaped, and sometimes you can see the nucleoli in them. Okay, so this is how you have to differentiate different WBCs in the smear. This is another diagram showing a neutrophil, eosinophil, a lymphocyte, a monocyte, and a basophil. Okay, so this is how you have to count the 100 cells. Okay, so start from one end of the selected field, you have to count 10 cells in a row. Okay, so then you move one field below and start counting the cells in opposite direction. Okay, then again you come one field below, then again start counting 10 cells in opposite direction. So like this, in this zigzag manner, you have to count 100 WBCs, then you have to uh, count them in percentages. Okay, so this is how you have to enter all the 100 cells uh, in a tabular manner. Okay, so then you count how many neutrophils, how many lymphocytes, how many monocytes, how many eosinophils or basophils, depending on their features and characters. So normally 50 to 70% of the cells or WBCs are neutrophils, 1 to 4% are eosinophils, 0 to 1% is basophil, 20 to 30% are lymphocytes, 2 to 8% are monocytes. So usually the differential leukocyte count is done uh, to identify whether there is a neutrophilia or eosinophilia or basophilia or neutropenia. Okay, so usually neutrophilia is seen in acute bacterial infection, whereas eosinophilia is seen in parasitic infection or helminthic infection or allergic reaction. Basophils are usually seen in allergic reaction, whereas lymphocyte count and monocyte count, they are going to increase in uh, chronic infections and chronic states of inflammation. Then absolute count, it is the total number of particular type of WBC present in 1 cubic mm of blood. Suppose the total WBC count is 10,000 per cubic mm of blood, then absolute count is equal to differential leukocyte count divided by 100 into total leukocyte count. Then you have to report the differential leukocyte count. So where uh, you have to write my differential count and absolute count uh, are the neutrophil, so and so. So like that you have to write the differential leukocyte count of all the cells. So this is in brief about how do you perform differential leukocyte count. Thank you.